Jai Shri Gurudev. In this class, we are going to discuss the animal physiology. Each and every organism has its own physiology in it. The physiology and the anatomy. The anatomy means various internal organs are present in each of the animals as well as in plants. In the animals, various internal organ systems are present. For say, the digestive system, the respiratory system, the circulatory system, nervous system, endocrine system, the reproductive system and the excretory system. All these internal organ systems are interconnected and they depend each other to perform one particular function. And each and every of the organ system has its own function to do. And these functions are studied in a scientific manner, and that is known as a physiology. Physiology, physio means the nature, the natural form of the function of the cells, the tissues or the organ systems are studied. If the study is done with the animal system, then which is called the animal physiology. Let us come to the definition for this animal physiology, which states that the scientific study of functions and vital processes of living organisms and the individual organs, systems, tissues and cells of which they are composed of. It is this scientific study where we study the functions and the vital processes happening in the living organism. If we study the functions and the vital processes in particular with the animal system, then we call it as animal physiology, where we study the functions of individual organs as well as the tissues, or in some of the cases which could be the functions of the cells, which all belong and perform the vital role in the living organism. That is, if we study which is in the animals, which is known as animal physiology. Always, all the animals, all the living creatures, here the animals are living in particular environment, and there will be coping up with the environment take place between the animal and the outer environment. In particular, the environment, the coping or the balancing or the equilibrium taking place between the internal environment with that of the outer environment. The internal environment means the body fluid concentration, the pH of the body fluid, the salt concentration or the ion concentration present in it, the temperature, the pH, whatever. These are the internal concentration or comprises the internal environment, and the animal which lives in a particular environment which become the external environment. And the factors, for example, the temperature within the body, the internal environment, and the temperature in the outer environment are always balanced, or coping nature will be there between the organism, animals or the plants with the external environment. This is known as homeostasis. The homeostasis, the word is first coined by Walter Kennan, which means homeo means same or similar. Stasis means standing or the equilibrium. In terms of equilibrium, maintenance of constant environment is known as homeostasis. Let us look into the definition for homeostasis, which says maintenance of the dynamic equilibrium. Maintenance of dynamic equilibrium. What for? To achieve a constant or nearly constant or nearly constant state of internal environment. That means in order to cope up with the outer environment, the animal attain or achieve a constant or nearly constant state of internal environment. For example, the body temperature, the fluid concentration, body fluid concentration, which include the plasma of the cell, the plasma of the blood, in which various salt concentration. Likewise, all the factors are maintained in a constant way or in a nearly constant state with that of the outer environment, and thus phenomenon is known as homeostasis. Okay, when we say the homeostasis, let us study with various examples for homeostasis in this class. That is the homeostasis of water, that is balancing of the water content, balancing of the salt and the glucose. And these are the three examples we are going to study in this class. Before getting into this, we must know some of the concepts, that is osmosis. As we familiar with the definition of osmosis, which says the movement of the molecules from the region of its higher concentration to the region of its lower concentration through semi-permeable membrane. This is the definition we have by hearted. We have dealt this definition, that is movement of the molecules from the region of its higher concentration to the region of its lower concentration, is known as osmosis. I am going to deal the other definition for that, that is similar to that but slightly different in its concept, which says the process by which molecules of solvents, not just the molecules, molecules of the solvents tend to pass through semi-permeable membrane from lesser concentrated solution into more concentrated solution till the equilibrium attains. This is known as the osmosis. Once again I repeat, the process by which molecules of these solvents tend to pass through the semi-permeable membrane. Till that you don't have any confusion. Through the semi-permeable membrane from a lesser concentrated solution into a more concentrated solution till the equilibrium is attained. In the previous definition we have stated that the movement of the molecules take place from the region of its higher concentration level, to the region of its lower concentration level through this semi-permeable membrane. Here also have written in a different way. That's it. Let us understand what do you mean by the solution. As we know, as we taught in the various chemistry classes, solution is the mixture of solvent and solute. Mixture of solvent and a solute become solution. Right? And in various terms the solution can be categorized, that is hypertonic solution, hypotonic solution and isotonic solution. These types of solutions are made in terms of the concentration of the solute present in the solvent, that is the solute present in the solution. Let us assume there are systems A, B and C. There are three systems called A, B, C present. In the system, I take 1 liter of water, that is the universal solvent. In each of the systems, 1 liter of water is taken. And to the system A, I add 100 grams of salt, any salt, 100 grams of salt, that become the solute. To the system B, I add 200 grams of salt. To the system C, 300 grams of salt. Now we can say A has now become the solution, B system the solution, and C simply A, B and C are the three different solutions. Three different solutions we would say because in each of the solutions the salt concentration is varying, the solute concentration is varying. But how can we distinguish these three systems as hypertonic solution or a hypotonic solution and hypertonic solution? That is the question. In order to understand or in order to differentiate the systems or the solutions, the kinds of solution, we must compare the given solution with that of the other solution. Then only we can say the given solution is hypotonic or isotonic or a hypotonic solution, like that. Let me take another system D. In that also the solvent is 1 liter of water, and to that I add 500 grams of salt. 500 grams of salt. Let us compare system A and system D. In the system A, the solute concentration, if we take, for example, if you take 100 ml of solution from A and 100 ml of solution from D system, the mixture, in the 100 ml of A solution has lesser amount of salt when which is compared with the 100 ml of solution taken from this system, right? Then in that case, the solution A is hypotonic, and the solution D is hypertonic. So we can say solution is hypertonic solution or hypotonic solution. When we compare two systems together, one become the hypotonic solution, another become the hypotonic solution, which depends on the amount of solute present in it. So hypertonic solution and hypotonic solution. Likewise, if we compare system or solution B with that of the D, then also the B system become hypotonic solution and D become hypertonic solution because the salt concentration is more in solution D. The similar way C become hypotonic solution and D is hypertonic solution. Suppose if I add to 1 liter of water and 300 grams of salt to it. Now the D is a solution. Let us compare the A and D system. The A is hypotonic solution and D is hypotonic solution, right? B is hypotonic solution and D is hypotonic solution. But when we compare the system or the solution C and D, and both are having same amount of salt in it, right? In that case, the solution C and D are called isotonic solution. So the concentration of the solute between these two are similar. Therefore, these two are isotonic to each other. But when which is compared with other systems, that may vary, which could be a hyper or hypotonic solution. Let us move another example. Suppose if the D system is added or the solvent is added with 200 grams of salt. Let us compare A with the D. If we compare solution A with the solution D, the A solution is hypotonic and D is hypertonic. But when we compare B solution with D, both are having same amount of solute. Therefore, in this case, the B and the D are isotonic solutions. When which is compared with the C and D, the C has more amount of solute in it. Therefore, this particular system now become hypotonic when which is compared with the solution having 200 grams of salt in 1 liter of solvent, right? Suppose if we take 100 grams of salt in it, in the solution D, and compare to this, the A and D become isotonic and B and C become hypertonic when which is compared with D. That means when which is compared with B, this is hypertonic and this is hypotonic. And similar way, C is hypertonic and D is hypotonic solution. But the problem is, in order to understand the process of osmosis, we must know the movement of the molecules. The definition says the molecules are moving from the lesser concentrated solution to the more concentrated solution. Suppose if we consider the system A and B, here the A is lesser concentrated whereas B is more concentrated solution. We have to clarify with this is hypotonic. When we compare the system A with that of the B, this is hypotonic, this is hypertonic solution, right? Hypotonic and hypertonic, when these two systems are compared, in the hypotonic solution, the solute concentration is less when which is compared with the concentration of a solute in system B, where which is high. The other thing we have to look into is the water molecules. When we say it is a hypotonic solution, means when which is compared with the B, which has a lesser solute concentration, whereas high water concentration. Don't get confused with this. In the hypotonic solution, the solute concentration is lesser than that of the solution B, whereas water concentration is higher than that of the solution B. Therefore, in the solution, that is hypotonic solution B, the water is less, right. The definition says that the movement of the molecules of the water take place. When we differentiate the types, the solution into different types, hypotonic, isotonic, hypertonic, we refer with the solute. But when the moment we say the process, that is osmosis process, that is the movement of the molecules, we will refer with the water molecule here, and the molecules are moving from one system to other system, one environment to other environment, one solution into other solution, where a lesser concentrated solution, from a lesser concentrated solution into a more concentrated solution. Here B is more concentrated solution, hypertonic solution, and A is hypotonic solution. The movement is from lesser concentrated solution into the more concentrated solution. Here the lesser concentrated solution is A and more concentrated solution is B. Therefore, movement takes place from A to B, right. Because in a less concentrated solution, there will be more amount of water molecules present. Therefore, this excess of water get moved from this system into a less content of water present, that is the hypertonic solution. And this happens between the semi-permeable membrane, and this is known as osmosis. Therefore, the movement of the molecules, what molecules? Solvent molecules, not the solute molecules. Solvent molecules tend to move through semi-permeable membrane from one region to another. The region is less concentrated solution where the higher concentration of the water is present, to where the high concentrated solution or more concentrated solution where water is less. The other definition holds good now, that is the movement of the molecules. What molecules? The water molecules from the region of its higher concentration to the region of its lower concentration, one and the same. This is how the molecules are moving. Once again, the osmosis is of two kinds, end osmosis and exosmosis. Suppose if we consider this as a living cell, and thus in the living cell there is a fluid content present, the cytoplasm of the cell present, or which could be the plasma of the blood. So say in toto we call it as body fluid of the cell, and there will be interaction with the outer environment. For say this particular cell, let us consider this cell as amoeba cell, and which lives in freshwater form. That is the outer environment. Due to the various metabolic activities, there will be accumulation of metabolic products in the body fluid. Therefore, the body fluid become hypertonic solution which is compared with the fresh water, that is the fresh water become hypotonic solution. Clear? body fluid is hypertonic and fresh water become hypotonic. As we know the definition for osmosis, the movement of the water molecules take place from the lesser concentrated solution to the more concentrated solution. Hypertonic is the more concentrated solution. The fresh water, the outer environment is the lesser concentrated solution. Therefore, water molecules move from outside the outer environment into these cells. Water molecules move from outer environment into the cell. This through the semi-permeable membrane. This type of osmosis is known as endosmosis. Endosmosis. Endo means within. The movement of the molecules are taking place from the outer environment into the system. Endo means within the system. Therefore, this type of osmotic movement is known as endosmosis. Suppose in case of sea water, that is marine water, the animal's body fluid, in the marine water, the concentration of the solute is much higher than that of the body fluid. In that case, the marine water become hypertonic. Hypertonic solution. Right. Then the body fluid become hypotonic. So, the movement will be from the lesser concentrated, the hypotonic, lesser concentrated to the region of higher concentrated, more concentrated. Therefore, whatever the water molecules present in the system, that is present in the body fluid of the animal, from the lesser concentrated region where the water molecules are in higher, and water molecules move out of the system, that is into the outer environment through the semi-permeable membrane. Here the semi-permeable membrane is the plasma membrane of the cells or the animals, and in such condition the type of osmosis is known as exosmosis. Exo means outside. The movement of the molecule taking place from the cell into the outer environment. Therefore, the movement, the osmotic movement of the molecule is now the exosmosis. With this clear understanding, we will discuss some of the examples for water balance and salt balance, and as well as in another example, that is glucose metabolism. Never ever get confused with the statement. Here, when we talk about the movement of the molecules, we look into the concentration of the water molecules, which is higher and which move from the higher region to the lower region. But when we talk about the type of a solution, we look into the amount of the solute present in it. Therefore, the movement is decided by the solute concentration or the type of the solution, or always the movement of the water molecules are from the less concentrated solution to the high concentrated solution. Is that clear? We will discuss the water balance, salt balance and glucose balance as an example for homeostasis. Okay, let us study the homeostasis as water balance. One of the examples for homeostasis, that is how the water balance taking place between the outer environment with that of the animal. animal cells or the body fluid. In case of freshwater forms, the freshwater, the animals are living, the aquatic animals live in freshwater called freshwater animals. In that case, with that previous knowledge, we can say the body fluid is hypertonic. Right? The freshwater, the media, the outer environment become hypotonic. And we know the definition for the osmosis, that is movement of the molecules, solvent molecules take place from the less concentrated into the high concentrated region. That means here in the fresh water, the water molecules are high in number than the water molecules of the body fluid. Therefore, the molecules, water molecules are from the hypotonic solution, that is the freshwater outer environment, into the cells of the animal, and this is known as endosmosis. Correct? Endosmosis taking place. If the endosmosis is keep on happening in the system, the animal cannot survive. Suppose this is the system, the water molecules keep on entering, means the animal cell or the individual, for example amoeba cell, a single cell cannot withstand the pressure, right? In order to cope up this or come out of this problem, for example, in case of amoeba, there are contractile vacuoles present, and these contractile vacuoles collect the excess of the water whichever come into the system through endosmosis, and the excess of the water will be eliminated out of the system, that is out of the cell, by the aid of the function of contractile vacuole. Thereby the excess water will be given out and the balance will be there, the water balance will be there. Likewise, in case of various crustaceans, there are specialized glands present called green glands, and these green glands absorb more water. The excess of water will be absorbed by these green glands and eliminated out of the body. That is how the crustacean withstand this endosmosis condition. The freshwater fishes having the kidney, glomerular kidney, and where the filtering of the material take place, the excess of the water which will be eliminated out of the body. This is taking place in case of freshwater animals. In the freshwater animals, body fluid is hypertonic and the freshwater, the outer environment become hypotonic. Therefore, more and more water molecules enter from the outer environment into the internal environment, where the accumulation of the water molecule take place. In order to avoid that, in case of amoeba, contractile vacuole eliminate excess of water, crustaceans, green glands involved in that, and a freshwater form, the glomerular kidney involved and eliminate excess of the water. The similar way, when we discuss the marine animals, in the marine animals, the salt concentration is high in the water, that is, this become hypertonic. The outer environment become hypotonic, whereas in case of freshwater form, the outer environment is hypotonic. The solute concentration is less here, whereas in the salt water, solute concentration is high. Therefore, the salt water, the outer environment become hypertonic solution, whereas the body fluid become hypotonic.
tonic, right. Therefore, the osmosis taking place between the body fluid of the animal and the animal and the outer environment. The molecules move from lesser concentrated solution into the higher concentrated solution, that is from the body fluid, from the body cells into the outer environment, that is called exosmosis. So, due to which more and more water molecules are eliminating out of the body. In that case, animal will be dehydrated. Dehydrated means water will be eliminated, animal will be desiccated, dehydrated, low amount of or less amount of water present in the animal. In that case, in order to compensate that, the compensation of the water loss will be done by drinking of the sea water. More and more water will be drinking, out of which absorb water molecule, or out of the marine food, engulfing the marine food, in that also some amount of the water will be taken. That is how the lost water will be compensated. In the similar way, in the land animals also, dehydration will be there. The excess of the water loss will be compensated by the animal by showing two behavior, that is drinking and having food. Drinking of the water directly compensate the loss of the water, and feeding over the green plants. In the green plants there will be some amount of the water present, and that is also reabsorbed by the kidney tubules, and thereby which compensate the water loss. This is how water loss will be compensated or balanced. Homeostasis of the water will be taken care by the cells, group of cells, or the glands, or the organ, the kidney, whichever present. That is how which balance the water between the internal environment of the animal with that of the outer environment. We have studied the homeostasis of water. Let us study the balance of the salt between the internal environment of the animal with that of the outer environment. In case of marine animals, in order to compensate the water loss, they continuously, that is spontaneously, they are engulfing water, drinking water. More and more drinking of water compensate the water loss, but in the meantime there will be more and more accumulation of salt also taking place in the system, because the animal is drinking hypotonic solution, that is high solute concentration is present in that sea water, marine water. Therefore, again this become a problem for the animal, somehow which balance the loss of water, but along with that accumulation of the salt take place, therefore which will become lethal to the animal, danger to the animal. In order to take care of that problem, in the marine forms there will be a removal of excess of the salt take place. For example, in the marine fishes, in the gill region there are specialized cells present, and these cells are called chloride secretory cells. On the margins of the gills, special type of cells present, and these are called chloride secretory cells. And these cells absorb more amount of the chloride content, the salt content present in the sea water, and which will be eliminated out. Therefore, animal is devoid of high salt concentration, and water will be balanced for the water loss. Likewise, in the sea turtle and the marine birds, they tend to drink more and more water, since the turtle live within the sea water itself, and the sea birds are having food in that locality, habitat itself, or in some cases which also take amount of water along with the seafood. In that case, more and more salt will be accumulated in the turtle as well as in the birds. Therefore, the salt concentration is higher in that case. In that case, in the head region of the turtle and the marine turtle and the marine birds, there is a specialized gland called salt glands. These salt glands absorb excess of the salt and eliminate it out of the body. That is how which balance the salt. When which comes into the freshwater forms, in order to eliminate more and more salt content, the animal pushes the water. There will be water loss, and along with the water, more and more salt also get lost out of the system. In that case, the freshwater forms like fishes and the crustaceans, there are specialized cells present, chloride cells in the gills or the kidneys reabsorb the amount of salt required for the system. While the excreting out, the amount of salt will be required form of amount of salt will be reabsorbed by the chloride cells which is present in the gills, as in case of crustaceans and the fish, or in the fishes there are kidneys. In the kidney tubules, reabsorption of the salt take place, which compensate the salt loss. That is how which compensate the salt or water loss or balance the water and the salt between the outer environment with that of the internal environment. This is another best example for the homeostasis. The other best example for the homeostasis is the glucose balance. Or glucose homeostasis. As we know, the normal blood glucose is 80/120 mg per 100 ml of blood. This is the normal blood glucose in a normal person. Always there will be elevation of the blood glucose level, and there will be a dropping of the blood glucose level happening now and then. But somehow our body is maintaining the blood glucose level in this range, that is 80 to 120 mg per 100 ml of blood. This is possible because of the endocrine system, that is the pancreas. In the pancreas acts as endocrine and exocrine. The endocrine part, the islets of Langerhans are present, and these islands of the cells possessing two important kind. There are 4 different types of cells present. In that, 2 important kind of cells involving in the glucose metabolism, those are alpha cells and beta cells. With this explanation, I will move on to the homeostasis of the glucose. And as we know, the normal range is between 80 to 120 mg per 100 ml of blood. Soon after the intake of the food, after having the food, the level of the blood glucose is going up, and the higher concentration of the glucose in the blood leads to a lethal condition, in a prolonged condition, which is hyperglycemia. Suppose if the glucose level is lesser than the normal range, the condition is known as hypoglycemia. Okay. The normal range is between 80 to 120 mg. If the glucose concentration is higher than 120 mg, there is a normal range, above this normal range, which leads to a condition known as hyperglycemia. Suppose if the blood glucose concentration is going down or dropping down than this range, then the condition is known as hypoglycemia. In a healthy human being, soon after the intake of the food, the blood glucose level increases. So, there will be a rise in the blood glucose. So, in that condition, the pancreatic cells, that the islets of Langerhans, in particular the beta cells of the pancreas, which get activated and which give a proteinaceous secretion, a hormone known as insulin hormone. Insulin hormone. This particular insulin hormone convert excess of the glucose which were present in the blood into a glucose is a monosaccharide, and which will be polymerized into polymer of glucose, that is polysaccharide glycogen. And this particular glycogen is get stored in the liver majorly and the muscles. In the muscle cells and the liver, excess of the glucose is converted by the activity of insulin into glycogen, and which get stored up in the liver and muscles. The process is known as glycogenesis. The generation of the glycogenesis take place. This is happening in the normal human beings. In the abnormal condition, for example, in the hyperglycemic condition, in an abnormal state, some amount of excess glucose will be converted by the activity of the insulin, which is given out by the beta cells of the pancreas, and through the process called glycogenesis, some amount of the glucose get converted into glycogen and stored in liver and muscles. Suppose there is a deficiency for the insulin hormone, and some amount of the excess glucose get eliminated through the urine by the activity of the kidney tubules. The kidney cells are active, and excess of the glucose will be filtered and eliminated along with the urine. Urine having glucose, and this condition is known as glucosuria. Glucose and urea, mixture of glucose with the urine, and this is a pathological condition in which excess of the glucose will be eliminated along with the urine. In a normal condition, urine is devoid of glucose. In an abnormal condition, excess of the glucose is eliminated, and this condition is known as glucosuria. This is happening when the normal range is exceeding, normal range of the blood glucose is exceeding. In that condition, this process are happening. Suppose if the person undergoes a starvation or they fasting, there will be no intake of the food. In that condition, a person going to hypoglycemic condition. In the long term, the person undergo a pathological condition known as hypoglycemic condition, or in a fasting or a starvation time, the body demand the glucose for the energy. In that case, the stored glycogen will be converted into glucose. In the starvation condition, there will be dropping down of the glucose. Therefore, the glycogen, the stored form of the glycogen, get converted into glucose. For that, there is another cell known as alpha cells. of the pancreas. Alpha cells of the pancreas give out a secretion which is known as glucagon hormone. Insulin is one type of hormone, and then alpha cells give out a glucagon hormone which involve in the process called glycogenolysis. Lysis of breakdown of the glycogen, a polymer of the glucose will be broken down, and the monomer molecules, that is glucose, will be given out into the blood level. In that condition, the blood glucose level reaches from a lower to the normal condition. Or in the normal condition, I told you, along with the urine, some amount of glucose also there is a chance for elimination. In that case, the kidney tubules reabsorb the excess of glucose and given back into the bloodstream. Therefore, blood glucose level is maintained. In another condition, that is in a stressed condition or in an emergency condition, the adrenal glands present on the top of the kidney. Over the kidney there are adrenal glands. This particular adrenal gland secretes a hormone called adrenaline. This is secreted in a stressed condition or emergency condition, adrenaline, which also act on the liver cells and muscle cells and break down the stored form of glycogen and give out the glucose, and glucose level is maintained or elevated. In this condition, which is elevated, or in a normal condition, the glucagon hormone involved in this process, that is conversion of the glycogen into glucose. This is known as glycogenolysis. Gluconeogenesis, the process of conversion of amino acids into glucose, that is protein form source into carbohydrate source, which is known as gluconeogenesis. New source genesis. By these three processes, that is glycogenesis, glycogenolysis, glucogenesis. With these three processes, with the involvement of two major hormones, that is insulin and glucagon, and another hormone and the emergency hormone, that is adrenaline, the blood glucose level is maintained, and this is known as blood glucose homeostasis or glucose balance.